So in this lecture, I'd like to talk a bit about colour vision, and that's the sensitivity that we have to the spectral content of light. So remember that light is made up of photons, which can be thought of either as particles or little waves, and the waves can have different wavelengths or frequency, and depending on the wavelength that they have, we perceive them as different colours. So roughly speaking, things that we perceive as blue have got shorter wavelengths, and things that we perceive as red have got longer wavelengths. So our colour vision is really this sensitivity to the distribution of energy at different wavelengths or frequencies in the light entering our eyes. So how do we achieve this sensitivity to spectral content? So why would you want to achieve this sensitivity to the spectral content of light? Why would that matter? So look at this photograph of some flowers. The top one is the original photograph, and the middle one has been processed so that it has lost all the luminance information. The dark bits have been made lighter and the lighter parts have been made darker, so that in theory this photograph now has constant brightness all the way across it. I say in theory because depending on the particular display you're looking at, inevitably that won't be exactly true. But it's still got the colour information, as you can see. Now down at the bottom on image C, we've kept the luminance information, so we still have the pattern of light and dark across the image, but now we've taken away all the colour information. It's just a black and white photograph. And you can see that it's now very difficult to distinguish the two types of flowers in the image that only has luminance information, whereas it's still very easy to do so in this image that only retains the colour information. So basically colour can be a really helpful way of discriminating between different sorts of objects, and in particular along some really important dimensions. So for example, is this blackberry ripe or not? Is it a nice dark purple colour or is it still red and will taste bitter? You can see that having colour information could really help with that. And there's suggestions that colour information may be evolved to be useful to us in social situations, to detect changes in blood flow in people's skin, for example. And while that seems somewhat far-fetched, there's actually some really nice evidence showing that humans are incredibly sensitive to small colours in the changes of people's skin that signal the amount of oxygenated and deoxygenated blood, the amount of beta-carotenes, how healthy people are, how healthy their diet is, whether they're sick. All this information that could be really critical to a social organism such as ourselves. And I personally find that quite a plausible reason why colour vision might have evolved. Okay, so that's why colour vision is useful, but how does it work in practice? Well, you remember that in your retina you have these cone photoreceptors, and humans actually have 3 different classes of cone photoreceptors with different spectral sensitivity. So that means they are more or less likely to absorb light of different wavelengths, and that's what's shown here. So you can see we have one class of photoreceptor, sometimes called the short wavelength, or the S cone, or sometimes called informally the blue cone. It's shown in blue here. This, as you can see, is most likely to absorb photons that are around 420, 430 nanometers wavelength, and they're really not absorbing anything beyond about 550 nanometers. And then we have 2 other cone types, the medium, shown in green, and the long wavelength cone, shown in red, that are both tuned to rather similar wavelengths, peaking at around 550 or 580 respectively. I want to point out that these cone sensitivities are very broad, so it's not the case that the M cone is only absorbing light at 550 nanometers. You can see it's absorbing light all the way from 380 to about 660 nanometers. We're just talking about its peak sensitivity. Now in this graph, the sensitivity has been normalised, meaning that they all peak at 1. But actually it may be more helpful to bear in mind this slightly different way of plotting that data. So here we've plotted the unnormalised curves, and actually this is plotted in terms of quantal efficiency. So the probability that if a photon hits a cone, it causes an isomerization and therefore a current. So that, in other words, the photon is detected by the photoreceptor. And you can see that the S cone, it peaks at shorter wavelengths, and actually it's far less likely to absorb a photon than the other 2 cone types. It might be worth also pointing out, do you remember how we were talking about the encoding of orientation as an example of population encoding? Well, this is somewhat similar in that here we're encoding spectral information. So what is the wavelength of light? And just as we were talking about what is the orientation of a stimulus, we could ask here, what is the wavelength of light? Only, of course, it's even more so than for orientations, it's possible that many wavelengths may be present at the same place on the retina. And you can see that this is an example of really coarse encoding. So even though we've got loads of different wavelengths, all the way from 380 to 700 nanometers around in the visible spectrum, all that rich information is being encoded with only 3 classes of sensor, with this really broad tuning and this overlapping spectral sensitivity. So you can see that we're going to lose a lot of information about the wavelength present in the light just because it's encoded with so few sensors. The encoding is not uniform across the retina either. So this is an amazing image showing the retinal cone mosaic in an individual human observer. The cones have been colour-coded. They're not actually this colour, it's not like they appear different when you examine the eye, but their sensitivity has been deduced and they've been colour-coded so that you can easily see which is which. So the red ones are the long cones, the green ones are the medium M cones, and the blue are the S cones. And this is the fovea in the middle. So you can see that in the fovea the cones are very dense. You'll also notice that there are no S cones. So right in the middle of our fovea we don't have any S cones. And even around outside the fovea you can see that the S cones are far less common than the L and the M cones. So to go through that in a bit more detail, in this slide the green curve here shows the spectrum of light from a particular surface. So it might be light reflected off a wall painted in a particular colour, maybe a sort of greenish colour. And this is the spectrum of light. So you're getting a lot of light at 550, a little bit at 420-ish, and then, less in between, less around 600, and then a bit more at longer wavelengths. Okay, and this is what's called a broadband spectrum, because as you can see it contains light of lots of different wavelengths. It's not the case that there's just one wavelength of light present. And then to work out how each cone responds, you have to do this mathematical operation here, which I've written with these integral signs. But actually it's very similar to the spatial receptive fields that we were talking about earlier in the context of simple cells. Basically, for each wavelength present in the stimulus, you say, okay, how much of that wavelength does the image have? This is this value I as a function of lambda. And then you say, how sensitive is the cone type? And these are these curves here. And then you multiply those together. And then you go to the next wavelength, like 402, let's say. Again, you find out how much energy the spectrum has of that wavelength, how sensitive the cone is, multiply the two together and add them up and keep on going. And you do that over all the wavelengths that are present in the image. Obviously for the S cone you wouldn't have to go much beyond 550, because then you would find that this absorption would be 0 anyway, so absorption for the S cone. So you do that, and then each cone type is then just giving you a single number. So this entire complicated spectrum has been boiled down to just 3 numbers representing the activity of each cone type. So you get, from this whole spectrum I lambda, you get 3 numbers, L, M, S, the activation of each of the cone types in that area of the retina. So that means that whatever the spectrum that's coming in on your retina, it just gets represented by a different set of 3 numbers. Now, interestingly, this isn't true the other way around. There are some LMS triples that can't be produced with any spectrum. So, for example, you can't get 0, 1, 0 because there is no type of light that can activate the M cone without also activating either the L cone. If the light's reddish over here, say 650, you can see that's going to activate L, but also M. Or it's going to also activate the S cone. So you might say, well, okay, let's go over here to say 450 nanometers. Now the L cone is not responding very much, so I can activate M without activating L. But then the trouble is you're activating S a lot. So again, you're not going to get 0, 1, 0 for your cone type. And it's an interesting question to ask. If we could somehow artificially activate only the M cones, what colour would we see? Would this be a new colour that humans have never seen before? I have no idea. I don't think anyone's done the experiment yet, although people have certainly talked about it and wondered if maybe you could use a very precise laser to target just the M cones. So you're hitting M cones without hitting the L and the S cones. What would people see? So that's a question you may know the answer to one day. Now you can see that if all possible spectra that we could ever see in the world can be represented as just 3 numbers, there are going to be many different spectra which yield the same 3 numbers. And those combinations of spectra which yield the same S values are called metamers. It's not a very helpful word, but a metamer is a physically different stimulus which creates the same physiological response and which therefore can't be distinguished